Welcome back to another important lecture. And since in the last class we started discussing the organelles that we have within the animal cell, and in today's class we would further look on toward two major cellular structures that we have within the animal cells, and among them we would be talking about the lysosomes. And then we would be discussing about the mitochondria that we have within the cells, and you know it very well from the previous classes that the mitochondria they are the powerhouses of the cells because they have to be generating the energy. Now, if I were to tell you something about the lysosomes, you see that as far as the life of the human is concerned, so this has got some limit. After some time, we die. So likewise, within the cells, we have got multiple organelles, and these organelles, after they have lived for some certain period of the time, so they have to be removed from the cell cytoplasm, and they have to be replaced with the new cellular structures. So we will see that how come these lysosomes that we have got within the animal cells, they would be contributing the role in that. Now, before I proceed, one thing that you have to keep it in your mind is that these lysosomes, they have got some enzymes within themselves, and these enzymes, they are the hydrolytic enzymes. And by the hydrolytic enzymes, what we mean is that they would digest some complex substances into the simple ones by the use of the water. Now, the idea is that if I were to have this cytoplasm, and in the cytoplasm, if I have got these lysosomes, and these lysosomes, they have to be enclosed by a membrane because we would never want that enzymes must leak out of the lysosomes randomly because if that would happen, then the animal cell would die. It would not be able to live. So that is why they are the membrane bound structure and the size of these lysosomes is 0.1 to 0.5 micrometers, so they are not that much of the big structure that we have within our cells. And if I were to tell you that how come these lysosomes they would be formed, so they would pinch off from the Golgi bodies and afterward they would be retained within the cell cytoplasm. And as far as the pH of the lysosome has to be bothered about, then you must know that the pH that we have got for the lysosome is 4 to 5. Overall, the cytoplasmic pH is near to the neutral, but since the lysosome has got these hydrolytic enzymes in it, so that is why the pH of the lysosome would be less, like in between 4 to 5. And if we just try to say that how much of the total of the enzymes we have got within these lysosomes, so they are more than 60 of the enzymes. And generally, they are referred to as hydrolases enzymes, like generally the enzymes that we have in the lysosome, so they are termed as hydrolases. And among the hydrolases, we have got these lipases that would break down the lipids. We have the nucleases that would break down the nucleic acids. Then we have the carbohydrates that would be digesting the carbohydrates, and then we have got these proteases enzymes within the lysosomes that would be digesting the protein components into the amino acids. Now, if I were to show you the diagram of this lysosome, so here you see that for this lysosome, on the outside, what we have got is this membrane, so that the enzyme must not leak out. And within the lumen of the lysosome that we have, so here there would be the presence of the enzymes. And another important thing is that within the cell membrane of these lysosomes, we will also have some transport proteins, and these transport proteins they would be moving some charged particles into the lysosomes that would be with an intention to be fluctuating or changing the pH of these lysosomes that we have got. An important concept is that we must be very much clear that how come these lysosomes they are being formed. Earlier I have said that they pinch off from the Golgi bodies. But the situation is that since these lysosomes they have got the enzymes in it, and enzymes are protein, and whenever the protein has to be formed, so the information must come from the DNA. So at any time, when the animal cell decides that, okay, let us make some lysosomes and we should send it or we should retain them within the cytoplasm. So some part of the DNA that carries instructions for making the lysosomes enzymes, so they would be copied on the messenger RNA. We would take the messenger RNA out of the nucleus and then we would take it to the ribosomes. On the ribosomes, the proteins would be formed, the enzymes would be formed, but these enzymes they are yet not modified. So after I have made them on toward the ribosomes of the rough endoplasmic reticulum, I would take these proteins now toward the Golgi body. And within the Golgi body, the enzymes would be modified and they would become some enzymes would become nucleases, some lipases, some proteases, some carbohydrates. And now the Golgi vesicle would pinch off having the modified enzymes in it, but this Golgi vesicle would not be allowed to fuse with the cell membrane. But rather than this Golgi vesicle would be retained within the cytoplasm. So now this has become the lysosome for us. Okay, so this is how the origin of the lysosome goes as. And if we further try to be talking about the functions that this lysosome is performing now for your IGCSEs, you may have seen a chemical process where the sperm cells that come from the male body and the egg cell that is produced within the female body, so they had to fuse with one another for the fertilization process. Now, the situation is that these egg cells, they have got a jelly coat in it. And until or unless we don't digest off the jelly coat, then the nucleus or the genetic information that we have brought from the male parent in the nucleus of the sperm cell, so they both can't fuse. So just the situation is that in the head of the sperm cell we have this acrosome, and this acrosome is a specialized kind of a lysosome that would release the hydrolytic enzymes out of it, and the jelly coat of the egg cell would be digested, and when this is digested, then the fertilization would be brought about by. The other scenario is that since I have said that the organelles within the cells of our body, so the organelles they get damaged or they end up or they complete their life cycle and then we have to replace them with the new organelles. So the situation would be that whenever the damage or the worn out organelles has to be removed out of the cell, so again the lysosomes would be coming into the play. Afterward, you see that when an infant is born or the baby is born, so the baby they have got the long bones, like the bones that we have in the arm and that we have in the leg. So they are the long bones. Initially, at the early age, when we humans are, so at the end of the long bones, we have got a major component of this tissue, and this tissue is cartilage, and this cartilage has to be replaced by the bones. So the situation that is created in the body is that the lysosomes they would digest the cartilage that is available at the end of these long bones, and afterward, this cartilage would be replaced by the bones. Okay, so moving onward, you see that at some point you people may have heard about the specialized type of the white blood cells that we have in our body, and these white blood cells they are entitled as phagocytes. Why we call them as the phagocytes? Because they eat up the pathogens. Now, the scenario is that by some mean or the other, some pathogen has entered in our body, and this bacteria has to be engulfed. And what is engulfing this bacteria up? So, this is a phagocyte with us. Okay, so at any cost, I would never want that if I had to digest the bacteria, so my phagocyte should die. So the situation is that initially this bacteria would make the contact with the cell membrane of the phagocyte. Afterward, this bacteria would be enclosed within a vacuole. And once we enclose the bacteria within a vacuole, so it becomes phagosome. Now you see that I just want to kill this bacteria, not the phagocyte. So here you see that these lysosomes, they are abundant within the phagocytes. And these lysosomes, they would fuse with the phagosome that have the bacteria in it. And by the fusion of the lysosome with the phagosome, now this structure becomes phagolysosomes. And once we release the hydrolytic enzymes out of these lysosomes, so this bacteria would be killed, this would be digested, and you see by doing so, we did not do any of the damage to the phagocyte, but only we have eliminated or we have killed this bacteria particle that got entered into our body. Now moving onward, you see that if we talk about our mothers, so they have the reproductive tract in them. Okay, like you know that the female individuals they have the ovaries and they have the oviducts and afterward they have got the uterus in them. So when the mother is developing a baby, so the uterus basically increases to an enormous size. And when the baby would be delivered, so the uterus has to revert back to its original size. So at that time again, you see that the lysosomes would be contributing their role. They would be excreting the digestive enzymes, and when those digestive enzymes would be released, then ultimately the some part of the uterus would be digested, and then as a result of that, the female uterus it would revert back to the original size. Now again, there happens to be a phenomenon that happens in these frogs, and this is the metamorphosis. Okay, initially the frogs they lay the egg, and out of the egg, like we would make at some certain point the tadpoles. Okay, and these tadpoles you see that they have caught the tail in them, and now it has to mature into an adult frog. So again, the lysosomes would be released, and these lysosomes they would digest off the tail of this tadpole, and then by that further it would revert into an adult frog. So these are all of the things that you have to remember in context of the lysosomes. Now you see here comes another important organelle, and this is not something new, okay? This is something or a cellular structure that we hear about or learn about from the very basic classes. And we know this is mitochondria. Now, the situation is that we organisms are living or we are surviving because within the cells, the energy is released. And by the release of the energy, you see that we are able to perform the work. So, this mitochondria is something that is releasing the energy. So we must quickly see that how this mitochondria is designed or looks like. So you see that the mitochondria, like the nucleus, is a double membrane organelle. It would have an outer membrane and then this would have an inner membrane in it. And then in between the outer and inner membrane, we have got the intermembrane space. Then we would have a fluid-filled space within the mitochondria, which is termed as matrix. So M for mitochondria, M for matrix, and we have got some small ribosomes within the mitochondria, and they are the 70s ribosomes. And then we have the circular DNA in them. You see, this is something very special. Because we know overall the cell has got the genetic material in the nucleus, and our DNA is not, you could say, circular, but rather than it is a linear DNA. And the ribosomes that we have got attached to the membrane of the rough endoplasmic reticulum, and that we have got in the cytoplasm, so they were the bigger ribosomes, ATS ribosomes. But this mitochondria is unique in a way that it has got 70S ribosomes and the circular DNA in it, which are actually the characteristics of the bacteria, but rather these happen to appear in the mitochondria as well. And other than that, in the inner membrane of the mitochondria, we will have some proteins with us, and these proteins they are termed as the channel and the carrier proteins. We need not to know much detail of these, it would come in your A2 biology. So if I were to show you the diagram of this mitochondria, so you see that this mitochondria, this is caught on the outside having its outer membrane, and the inner membrane that we have to be seeing here is, so it has got a lot of enfoldings in it. Okay, and the other name of this inner membrane is also termed as the cristae. Now, if you were to see clearly, within the mitochondria, we have got this fluid-filled space. And this fluid-filled space, this actually has some enzymes for one of the mechanism that happens within the mitochondria, and that is the Krebs cycle. So for the Krebs cycle, the enzymes are actually available here within the matrix. Okay, the other situation is that within the matrix again we have got this circular DNA of the mitochondria, and then we have got also these 70S ribosomes as well. So this means that since the mitochondria has its own circular DNA, so it would allow the mitochondria to replicate on its own without relying on toward the nucleus. And since it also has the 70S ribosomes, so this means the mitochondria would be in the position to be making its own proteins. Okay, now the scenario is that in these inner membrane or the cristae, we have got this specialized channel protein, and this specialized channel protein is what we entitle as, or we call this as ATP synthase channel protein. And we will see in a while that this ATP synthase channel protein that we have, so this would allow for the making of ATP in the respiration process. And the last thing is that here we have this outer membrane and this was inner membrane. So in between them we have got this space. So the space between the outer and inner membrane is what we refer to this as an intermembrane space of the mitochondria. Now moving onward, the situation here is that how come these mitochondria that we have got, so they are able to be making or generating the energy. So you know it very well that in the mitochondria there would be a chemical process happening, and that chemical process goes by the name of the aerobic respiration. And in the aerobic respiration, what you know is that there would come a molecule of the glucose, and this molecule of the glucose would combine with six molecules of the oxygen gas. And as a consequence of that, we would make up six molecule of the carbon dioxide gas. We will make up six molecule of the water, and then the energy would be released in the form of ATP. Okay. Now we have to basically justify is that how come this mitochondria is able to make this energy in the form of ATP. Now the situation is that in your diet we are eating the sugars, we eat the proteins, and we eat the fats. And all of these biological molecule, they have got these elements in them, like we have the carbon, we have hydrogen, and then we have got the oxygen with us. Now, as far as this equation is to be bothered about, so I would talk with respect to the glucose molecule, that this glucose molecule has got hydrogen and it also has got some electrons in it. So whenever the energy has to be formed in the mitochondria in the form of ATP. So from this glucose molecule, we would be removing some of the protons and the electrons. And after we have removed them up, so I would take these protons and protons into the intermembrane space that we have in the mitochondria. And afterward, we would cause H positive ions to fall through the ATP synthase channel protein. And when the H positive ions would fall through the ATP synthase channel protein that we have in the cristae of the mitochondria, then we would be making ATP out of it. Okay, and wherever some work has to be done, so this ATP would be carried there, and when the ATP would have to release energy for some work, so this ATP would be broken down, and as a result of that, we would make ADP out of it, and then we would make phosphate out of it. Now, you see that this is ADP with us. And if I have to revert the ADP back into the ATP, so I would not use any other ATP molecule, but rather than I would take the protons and electrons from this biological molecule, and then we would again recombine the ADP with the phosphate, and the ATP would result out of it. If I were to show you with the help of the diagram, so if I were to say that you have eaten the glucose and the fats, so from these biological molecule, the protons, they would be removed off. And with a very complex mechanism that is a part of your E2 biology, these H positive ions they are accumulating, they are building up in between the outer and the inner membrane of the mitochondria. So what we could say is that these H positive ions they have accumulated in the intermembrane space. So when I have accumulated more of the H positive ion in the intermembrane space, so here the concentration of the H positive ion would increase, and in the matrix of the mitochondria, the H positive ion concentration would decrease. So now you see that these H positive ions, they are the charged particles, so they can't or they will not be able to cross these cell membranes directly, but rather than these H positive ions, they would pass through or they would fall through this ATP synthase channel protein. Again, for the moment, you just feel like that this ATP synthase channel protein is just like turbine, as in the turbines when the water fall. So the kinetic energy of the water is converted into the electrical energy. So likewise, anywhere in our body where we we perform the work and we reverted the ATP into this ADP and phosphate. So now this ADP and the phosphate they both would come to the ATP synthase. And the ATP synthase would say to these H positive ions that, okay, fine, you fall downward toward the matrix. So when these H positive ions, they would fall toward the matrix. So they would release energy out. And that energy would be utilized to be combining the ADP with the phosphate. And when they would be combined, so you see that we have made the ATP out of it. So the situation to be considering here at the moment is that in order to make the ATP from ADP and phosphate, I did not use any other ATP molecule, but rather than I took these H positive ion from the biological molecule, I allowed them to be falling through the ATP synthase channel protein. So this is a kind of an interconversion of energy, and at the end we have made this ATP out of it. Now, from the paper point of view, again, you could say that these mitochondria, when we try to look onto them with the help of the electron microscope, so they can appear in two ways. Either they would have a longitudinal shape, like when we would take the longitudinal sections of the mitochondria, they appear to look elongated. Okay, so this is the outer membrane. And the inner membrane that we have here, so this is the cristae that looks like here. And here we have got this fluid-filled space within the mitochondria, and this was referred to as the matrix. But overall, this is the longitudinal section that we have got for the mitochondria. Now, another structure that we see in the photomicrograph is that here we have got the membrane of the rough endoplasmic reticulum, and on the membrane of the rough endoplasmic reticulum, you see that we have got these ribosomes onto them. Okay, so this is the longitudinal section of the mitochondria. Now, if I were to show you the transverse section, so again you see that these are again the mitochondria with us. So the mitochondria, they can appear just like the spherical shape, or they could appear on the elongated shape. The thing would be that how come we are preparing the slides? Okay. So if I were to just mark the areas of the mitochondria, so here you see this is the outer membrane of the mitochondria. The inner membrane, like in the longitudinal section, so here we see the enfoldings of the inner membrane in the form of cristae. Now, the last thing is that how can we be able to know that what would be the longitudinal and how would be the transverse section? So you see that if I have this mitochondria with me, and if this mitochondria, if this is cut along the length, or the side is cut along the length, then the mitochondria would appear like this as here. But if I were to prepare a slide, and in that, if the specimen that we intend to look under the microscopes, if it was cut like this, so as a result of that, the mitochondria, they would appear like this. So I hope you would be clear with the structure of the lysosomes and the mitochondria. So see you in the next class.